Okay, good afternoon everyone, good afternoon. Okay, perfect. Good afternoon, good afternoon. So, okay. Good afternoon Shafia, good afternoon, good afternoon Archana, good afternoon. So friends, today now we are going to study the most important topic, that is the cancer biology. And I told to all of you that friends, the cancer biology is the most important topic with respect to the part B also, as well as with respect to the part C also. And friends, what kind of questions that we got so far from this particular cancer biology chapter? We all know that from the cancer biology chapter, friends, we can expect the questions majorly in the form of statements, the first one, and the second chance of asking the questions is the experimental based. Okay, there are only two ways of asking a question. One is the experimental based question. Second one is friends, statement related question. But 99.99% we can expect the question exclusively from this statement related only. So friends, I can tell you one important point. There will be no CSIR NET Life Sciences question paper without the question from this particular cancer topic. So that's why I am requesting every one of you, never leave even a small point in this particular cancer topic. The lighting is a little bit disturbing, just please try to understand. Okay, now focus. What we have to read exactly in this particular cancer biology? One second. What we people has to read exactly in the cancer biology? Friends, in case of a cancer biology, we have to read all the properties as well as all the characteristics. So focus everyone. We have to read first thing, the characteristics of a cancer, right? And friends, the second and most important thing where we can expect the questions is differences or the comparisons between the normal cell and cancer cell. And the third important thing which we have to study, the most important one friends, the differences between the benign tumor, then friends followed by the malignant tumor. And what about the fourth one that we have to study without fail? Is it echoing too much? Okay, try to rejoin Sumaya once we'll see it. Okay, so and the fourth one, we have to study the most important difference, genes involved here. Okay, genes involved is also very important friends. We people has to read it compulsory, the genes involved is. Now every one of you listen my statements very carefully without confusion. When we are reading, okay, is it too much echoing? Okay, can you hear me properly or is it too much echoing? Please let me know, is it too much echoing? Okay, please adjust for this. In the tomorrow class, I will set it up the volume, okay? Please adjust. So friends, we have to study the characteristics, we have to study the differences between the normal cell and cancer cell, we have to study the benign tumor and malignant tumor, we have to study the genes involved in the cancer. Only from these four areas we can expect the question. And the fifth possibility where we can expect the questions is the causes of the cancer. See, the word causes means the causative organisms. We know, right, certain bacteria can cause the virus, and certain bacteria can cause the cancer, certain viruses can cause the cancer. Whatever the bacteria and whatever the viruses that are causing the cancer, we will be calling them as the oncogenic organisms. So we have to read even about that. Friends, apart from these 5 areas, there is no way of asking a question. But whenever they ask a question, they will make the simple question. They will make a very simple question in a logical manner. So friends, it is very important to understand the given question correctly, and you have to choose a most appropriate option. Okay, now friends, we have discussed the cancer biology more than 2 to 3 times. So as it is a final revision, I will go little faster. If you have any kind of a doubt, you can immediately message me in the chat. I will go little bit faster. Okay, so now let's move on to it. Okay, so friends, how can we define the word cancer? Everyone listen to me. How can we define the word cancer? We all people studied many times that, friends, cancer is a genetic disease. I want every student has to listen this particular line, where most of the people will get confused. They will give a line that cancer is a genetic disease, but look at this, but it can be not inherited. Now how is that possible? We are calling that the cancer is a genetic disease, but why the cancer is not inherited from one generation to the another generation? What will be the reason? The reason is very simple. Follow me very correctly. I am calling cancer is a genetic disease. The only reason is it is caused by the changes in the gene sequences, or it is caused by the alterations within the specific genes. Because of the alterations or because of the changes within the specific genes, we are calling cancer is a genetic disease. And even though cancer is a genetic disease, 99% of the cases, friends, the cancer is not inherited. Now, how does this particular cancer will occur? We all people know that. Does any cell of our body can be affected by the cancers? Of course, true. But what's the point? You have to remember, 80% of the cancers will arise only in the somatic cells. What are somatic cells? All the body cells except the germ cells. I am requesting all of you, whatever the statement that I am explaining, every statement please try to remember. This should be a final revision. You no need to touch the book. So pay attention. So cancers are arising from what? Most of the cancers arise in the somatic cell. It doesn't mean that the cancers are not appeared in the reproductive cells. We have studied many type of cancers. An example, ovarian cancer. And friends, we have studied testicle cancers. So it is not that the cancers can only appear in the somatic cell, it never appeared in the germline cells, or it never appeared in the reproductive cells. But what is the point every student has to remember? The most probability of occurring the cancer when compared to the reproductive cell in the somatic cell is more. That is the point every student has to remember. Now friends, it is very important to remember the fundamental differences between the normal cell as well as the cancer cell, because friends, we can expect the questions majorly from where? Majorly from the differences only. So just a minute. Okay, now focus all of you. What are the fundamental differences between the normal and cancer cell? Everyone listen to my words very carefully. Friends, whenever we are talking about the normal cell, what are the simple characteristics performed by the normal cell? Everyone pay attention. Normal cell will undergo the normal division. What do you mean by the word normal division? Here the word normal division means, friends, regulated division. That is called as the normal division. And after division, do you think our normal cell can undergo the division continuously? Never ever. Our normal cell never undergo the division continuously. After a particular time of a division, our normal cell stops the division, undergo the process of differentiation. After differentiation, normal cell perform the function. Most importantly, once everything is over, the normal cell will die, which we can call, friends, apoptosis, which we also call as programmed cell death. If you can't remember all these points, remember two important points. What is the two important points? our normal cell never ever show the process of invasion. What do you mean by the word the process of invasion? Invasion means moving from one area to the another area. Suppose example, do you think our pancreatic cells can move from pancreas to the liver, or our liver cells can move from liver to the pancreas? That is not at all possible. What you have to remember, our normal cell never undergo the process of invasion. It never move from its desired area. And more important point, our normal cell never ever show the process of metastasis. What is metastasis? Forming a new colonies somewhere else in the body. That is called metastasis. That never be shown by the normal cell. Now, whatever the characteristics that I have explained for the normal cell, the cancer cell will show entirely opposite properties. Every student has to listen to me carefully. Follow me. The cancer cell undergo the division, uncontrollable division. And friends, now the question is very clear. Does the cancer cell will undergo the process of differentiation? I can use the word 99% cancer cell never undergo the process of differentiation. An example, some cancer cell can undergo the differentiation. Now can I say that will they undergo the complete differentiation? No, I can use the word that they will undergo the very low differentiation, or there will be a no differentiation process only. That's what you have to remember. Now, cancer cell undergo the unregulated division or uncontrollable division, never undergo the differentiation. What are the two major characteristics possessed by the cancer cell? That is, it always show the process of invasion. An example, my liver cell got infected with the cancer cell. My liver cell got infected with cancer. Now that infected liver cell can easily move into the other area, which means that every cancer cell can show the process of invasion. Now from the liver cell, that particular cancer cell, if it enter into the pancreas, obviously friends, it will undergo the process of metastasis. What is that? Developing the new colonies in that particular area. So friends, now all the students, please pay attention and try to understand the differences. Focus everyone. Look into this friends. The major differences between the normal cell and cancer cell, because nearly 2 to 3 times, I think 3 times, we got a question exactly with respect to the differences. Let's consider our 2022 February question paper. What kind of question we got? We got the question about the stem cell and the normal cancer cell. Correct? So most of the time they will raise the questions with respect to the differences, with respect to the statements only. That's why you have to remember every single statement. Please pay attention. Our normal cells are very small, uniformly shaped nuclei. Yes or no? Friends, how can we define the word tissue? We can say that group of cells, follow me correctly, group of cells having same size, having same shape, we can call tissue. Correct na? So you have to understand that all our cancer cells are very small. They are uniformly shaped. Never forget that point. But friends, if you look into the cancer cell, one cancer cell size is not at all same when compared to the another cancer cell. The entire size will be different. They are large, variable shaped nuclei. Please kindly don't get confused with that. And please friends, follow me. If you look into the normal cell, in case of a normal cell, cytoplasm occupy the larger position, or nucleus occupy the larger position. We all people know very well, undoubtedly friends, cytoplasmic volume is more when compared to the nucleus, but which is just the opposite in case of a cancer cell. What do you mean by that? Nucleus can occupy the lower position, and friends, cytoplasm can occupy the larger position. Don't get confused with that particular point. Now, if you look into the third one, very unique point, conformity in the cell size and shape. Once again, remember, conformity in the cell size and shape. What do you mean by that? Our normal cells having a fixed size, having a fixed shape, that is how they can form a tissue. But friends, if you look into the cancer cell, there is a variation in the cell size, variation in the cell shape. So what is the point I would like to highlight? Cancer cell actually never form the tissue. They never form the tissue. Why? Because they are not one cancer cell is not trying to associate with the another cancer cell, because their size is different, their shape is different. Friends, then they cannot form that particular tissue. And friends, another unique statement about the surface markers. This is also very important. On our normal cells, surface markers cannot be expressed every time. An example, let's take an example of a MHC molecule. What is the use of MHC molecule? MHC molecules are responsible for presenting the antigen. Yes or no? Now, you people try to understand, if there is no antigen, in our body, then what's the use of MHC? There will be no need of expression of that particular surface marker. But in the cancer cell, it never happened. Whether there is a use of the surface marker, whether there will be no use of the surface marker, the expression will occur continuously. That's what we have written here. Elevated expression of the certain surface markers. Always, whether there is a use of a surface marker, there will be no use of the surface marker, expression will be continued. Among all, the most important and significant point, lower levels of dividing cells and larger number of dividing cells. I can use the word that among all, this is the major point. Our cells are designed in such a way that it can undergo the division at a limited time, at a limited period. What do you mean by that? We all people know that, friends, how many number of times the cell has to be divided. It is genetically pre-programmed in our DNA molecule. Yes or no, friends? How many number of times the cell has to be divided? That many number of times only it will divide. If it is trying to divide extra, immediately our cell regulatory mechanisms comes into the picture. They will stop the division. But what about the cancer? There will be no regulatory mechanism, hence no one can stop the cancer cell to undergo the rapid. We don't use the word rapid division, we use the word uncontrollable cell division. Why we are using the word uncontrollable cell division? Because there is no regulatory mechanisms that is controlling the cancer cell. Simple point. So friends, these are the major fundamental differences between the normal cell as well as the cancer cell. Now friends, what is malignant tumor? What is benign tumor? Even we got the direct part C question from the malignant as well as the benign tumors also. See, I want every one of you understand the concept very clearly, what exactly this particular malignant or benign tumors. Just a minute. Okay, so all the students, please look into the diagram very carefully. If you look into the benign tumor, just follow me very correctly, friends. All the students follow me correctly. When I use a word benign tumor, please remember, the cells of the benign tumor, listen, they look like a normal cell. They function like a normal cell. Now you can ask me a question, then why you are calling it as a tumor? If it is looking like a normal cell, if they are functioning like a normal cell, then why you are calling it as a tumor? We never call a benign tumor as a cancerous. We always call benign tumor is the non-cancerous. There is no doubt about it. But what's the significant point every student has to remember? Then why we have used a word called as a tumor? We have used a word tumor, the reason behind that, they are all arranged in a group. They are arranged in a bulk amount. That's the reason only we have used a word tumor. Friends, look at this. How our normal cells will be arranged? Our normal cells will always arranged in an organized manner like this. very organized manner our normal cells will be arranged. But you only look into the benign tumor, how does the cells are arranged? The cells are arranged in a bulk like this. So there is no fixed position, there will be no fixed arrangement in the cells, which doesn't mean that, benign tumor doesn't mean that they are cancerous. Benign tumors, they just arranged in a bulk. That's why we have given the name tumor. We all people know, right? Some people on their hands, on their chest, on their back, they will get lipomas. What is lipoma? Lipoma means, friends, abundant amount of cells come close together and they will accumulate at one particular area. That is only we can call lipoma. So most of the people will think that when they observe on their hand, when they observe on their chest, they feel a bubble-like structure and they will think that it is a tumorous. It is not. It is a cancerous. It is not at all cancerous. It is a non-cancerous, which is the benign tumor. Now you can ask me, sir, will it cause any pain or can it be removable? It can be easily removable. ORR, if you don't remove throughout the lifetime, it won't cause any kind of a effect because they are non-invasive. They cannot move from one tissue to the another tissue. Don't get confused with that. Now, which is the most dangerous? Obviously, friends, the most dangerous one is what? Undoubtedly the malignant tumor. Of course, why malignant tumor is dangerous? The reason is very simple, two major reasons. One is it will undergo the process of invasion. Correct? Na? It is moving from one area to the another area. And the second reason why we are calling it is the very dangerous, friends, the simple reason because of the process of metastasis. Only these two reasons. Because of these two reasons only we will be calling malignant tumor is the cancerous tumors. Okay, so now let me explain you the major and the fundamental differences between the normal tumor as well as the malignant tumor. It is very important. I don't know what you people do. You have to remember all the statements without fail. Okay. Now, friends, focus all of you. Look into this. The benign tumor. It is a non-cancerous tumor. From here also we got the part C question. What tumor it is? Non-cancerous tumor. In the February 2022 we got one question. What is the question they have asked in the part B? How the cancers are differentiated. That's the question. So we can only differentiate the cancers, friends, based on which tissue they are arising. Suppose example, there is a breast cancer, there is a throat cancer, there is an ovarian cancer, is it? There is a pancreatic cancer. Correct? Now, based on what we have defined, because every cancer can show the similar kind of a properties only. So we have differentiated one cancer with the another cancer with respect to which tissue they are getting arise. That's the point you have to remember. Anyway, look at the differences. It is a non-cancerous tumor and friends, it can be removable. There is no doubt about it. Now, malignant tumor, it is a cancerous tumor. Now the question is very valid. Can this particular cancerous tumor, is it removable or not? Valid question. Undoubtedly, my friends, it is removable. But the point is what? When we have to remove it, before it undergo the process of a metastasis. If it undergo the process of a metastasis, what will be the use? See, there was a question asked in the CSIR exam. A person was diagnosed with the stomach cancer and he was treated successfully with the radiation and chemotherapy. After 6 months, once again the tumor was appeared in his pancreas. This is the 4-marker question they asked. Which are the following statements are the most suitable statements for this given question? So obviously what? That particular tumor from the stomach, it went to the pancreas. It has shown the process of metastasis. Which means, you have to understand, you can completely remove the cancer before it undergoing the process of metastasis. If it undergoes the process of metastasis, we can't cure. Even though we remove it, we can protect that particular area, but the cancer has spread somewhere else in the body. Now, benign tumors does not show the process of metastasis, as well as friends, it is non-invasive, which means that it cannot move from one area to the another area. Whereas it shows metastasis does invade other body parts. Now, all the students, you might observe, I am talking about the lipoma. Suppose some people will get the lipomas on their hand and on their chest. Whenever that, suppose example, on my chest I have one lipoma. Friends, even after 1 year, even after 10 years, the size of the lipoma is remain as it is. Okay, the size is exclusive, the size is same as it is before. But, so it stops growth after reaching a certain size, because which is the characteristic of a normal cell. Because normal cell never increases its size continuously. Normal cell, whenever it reaches a particular stage of maturity, it will stop its size. Correct? But which is not possible in a cancer cell. Everyone look into this. Malignant tumors shows indefinite growth, which means that friends, their size will be increasing continuously. So there will be no restriction to its size. That is the point I would like to highlight. Next, friends, this also very unique statement which was asked in the exam in the 4-marker question as a single statement. That is the adherence. What do you mean by the word adherence? Everyone just pay attention. This is one normal cell, okay? And this is another normal cell. Now you can see, right, both the cells are connected with each other. Yes or no? Of course, yes. Both the cells are connected with each other. Yes, no? Now the point you have to remember, the cell number 1 will restrict, listen, follow me very correctly. The cell number 1 restrict excessive growth of the cell 2. Don't confuse, friends. Similarly, cell 2 can restrict the excessive growth of the cell 1. So why is it happening? Why the cell 1 can control the cell 2 and how the cell 2 can control the cell 1? How is that possible? That is possible, friends, just because of the process of adherence. So what is the statement I would like to conclude? Adherence can inhibit the excessive growth of a cell. Very simple, but friends, do you think, can you observe any kind of adherence in case of a cancer cell? Now listen, you know what is the specialty of a cancer cell? Suppose this is one cancer cell. This is another cancer cell. Now understand what I am saying. When the cancer cell 1 try to come in contact with the cancer cell 2, you know what will happen? Cancer cell 1 will migrate, will jump. They will avoid the process of adherence. Once again, listen my friends, what they will do? They will avoid the process of adherence. By avoiding the process of adherence, they can grow indefinitely. They can grow uncontrollably. So please remember, there is no adherence among guest cells. They tend to slip, they tend to escape, they tend to avoid contact with each other. And what's the common point? The common point is it is less fatal to the body, and friends, it is more fatal to the body. Very simple statements. I don't know what you do. You people have to remember every single difference without fail. Now friends, what are the various types of cancers actually? Friends, according to you, how many number of cancers are there, friends? Please keep it in your mind, there are more than. You all people will think that there are 100 to 200 different type of cancers are there. Keep it in your mind, friends, there are more than 40,000 types of cancers are there. And the point is very clear, whether there is a 40,000 of cancer, whether there is a 20,000 of cancers, doesn't matter. All the cancers are differentiated exclusively based on their tissue of origin. Based on what, friends? Based on their tissue of origin, we have differentiated into different types. And why I kept only these? Why not any other? Why I have kept only these? Because, friends, these are all the most dangerous cancer, and most, instead of using the word most dangerous cancers, we can use the word most commonly occurring cancers. That's what you people can remember the statement. Bladder cancer, breast cancer, colorectal. Please remember, they can ask a Part B question. Which of the following type of cancer is the most commonly occurring cancers? Okay, so you have to remember it. Lung, lymphoma, melanoma. Friends, among all, see, just listen. All cancers are very dangerous, there is no doubt about it. In this particular table, which are more deadly cancers? Melanomas, lymphomas, as well as the brain tumors. Because, friends, the lymphoma cancers are what? They are actually affecting our B cell also, they are actually affecting our T cell also. We all people know very well, without T cell, it is impossible in our body, the B cell can produce antibody. Please don't confuse, even though B cell is normal, if the T cell got cancerous, if the T cell got mutated, it is impossible to synthesize the antibodies. We have to take the antibodies only externally. Our body cannot synthesize the antibodies on its own if there is a damage to the T cell. So that's why whether it is a B lymphocyte cancer or the T lymphocyte cancer is the deadly cancers. Then melanoma, which are a type of skin cancers. You don't know, friends, when we people exposure to the higher intensity of sunlight, what will happen? We have studied in the DNA repair mechanisms also. Pyrimidine dimer formation occur. If we don't treat that pyrimidine dimer formation, obviously what is going to happen? It causes the different type of skin cancers. Okay. Suppose example, if you look into the pancreatic cancer, there is only one. If we don't treat the pancreatic cancer in the early stages, there is only one treatment. What is that? The transplantation of the pancreas. But I told to all of you many times, nowadays because of technology, entire transplantations are becoming so successful. Whether it is a liver transplantation, kidney transplantation, or pancreas transplantations are occurring successfully, but the organ is not working like before. Even though after successful transplantation, the persons are not living not more than 1 to 2 years. So these are all what? Different type of cancers. Now every student, please look into this particular diagram very carefully without having any kind of a confusion. Just listen. 1 second, friends, 1 second. There is. Okay. So friends, please look into this diagram very carefully without having confusion. Normal cell, dysplasia, carcinoma, invasive carcinoma, metastasis. By looking at these 5 diagrams, I want to say one statement. What is the statement I want to give by looking at this 5 diagram? Cancer never spread very faster in the early stages. Once again, listen my statements very carefully. Cancer never spread very faster in the early stages. What do you mean by that? Because cancer cell will undergo the rapid division only when it undergoes the process of metastasis. Whenever it is moving from that particular area to the other area, then only it can show the super spreading, or then only it can show the uncontrollable division. All the students, please look into the diagram very carefully without confusion. This is a normal cell. The cell was not at all infected with the tumor. You all people can see here, all the cells are absolutely normal epithelial cells. Please look into the diagram. In a group, everyone focus. In a group of epithelial cells, there is only one cell. Imagine there is only one cell in that particular epithelial layer got infected with cancer. It is dividing uncontrollably, which doesn't mean that immediately it can transfer to all the other cells in that particular tissue. No. What is the reason? Please follow me very correctly, friends. Suppose example, there are 3 cancer cells are there. Do you think these 3 cancer cells immediately affect all other cells in that particular tissue into cancers? No. Because we are having several immune defense mechanisms. Our mechanisms, let's take an example, cytotoxic T cells, TC cells. What our TC cells will do? Our TC cells will try to destroy the particular cancerous cells which are in the early stages. So if we kill this particular newly growing cancer cell in the early stage, of course it can never spread. But if our immune defense mechanisms fails to kill that particular cells in the early stages, now you remember, it is spreading slowly and it is affected that entire tissue. In a tissue, all the cells are not at all affected. In a tissue, only few cells are affected. That few cells, very slowly, not rapidly, very slowly, they are affecting all other cells, which we can call the dysplasia stage. You know, if you identify the cancer in the dysplasia stage, we can easily treat that particular cancer. But what is the greatest disadvantage here? The greatest disadvantage, during the condition of a dysplasia, there will be no symptoms. There will be no symptoms at all. So due to the lack of any symptom, we people never experience any symptom. As a result, we never understand the tumor is growing or not. So from the dysplasia, it will spread the entire tissue. Now all the students, please look into this diagram, the most important diagram. Anyway, I'll explain it in the next slide also. All the students, please pay attention. Look into this. Now it has been affected the entire tissue. Friends, pay attention. It has affected the entire tissue. Once it affected the entire tissue, it has to move. Suppose example, this is epithelial cell. From the epithelial cell, this cancer has to move to the liver. But how? Do you think the cancer cell can directly jump from the epithelial cell into the liver cell? That is not possible. So what's the mechanism? The mechanism is very simple. They will use blood vessels. Correct, na? Every student, please look into the picture very carefully. They are using either lymph vessels or the blood vessels. They can use the lymphatic vessels, they can use the blood vessels, and they can move to the newer areas. Now, most of the students will get one very valid doubt. What is that valid doubt? Sir, from where this blood vessels has come from? You have to remember that cancer cell will start utilizing our own blood vessels. The cancer cell starts utilizing our own blood vessels and it's a normal cell blood vessels to transport. Suppose example, in certain cases, you know what is the specialty of a cancer cells? Cancer cells can synthesize their own blood vessels for their transport. So that particular transport only, we will be calling it as the angiogenesis. I'll tell you. Keep it in your mind. Angiogenesis is the process of transport of a cancer cell or the invasion of a cancer cell from one area to the new area. So let me explain you a little more elaboratively the process of the metastasis. After this we will discuss. Okay, this is the similar type of a question which we got in our 2022 paper. Just a minute. Okay, friends, listen. This is the same type of a question which we got in the exam, but in a different way they have asked. But the concept is same. Everyone pay attention. Cancer growth, follow me friends, often depends on defective control of the cell death or the cell differentiation. See that line? They will give the same line, they will ask us whether the statement is a true statement or a false statement. Always remember, absolutely true statement. See, once again look into this. Cancer growth depending on defective control of cell death. What do you mean by that? Our normal cells has to undergo the process of what? Apoptosis, which we can call programmed cell death. Now you all people know very well that every cell in our body, once the cell become older, what friends? Once the cell become older, it will undergo the process of apoptosis. There is no doubt. That is the biological process has to happen in our cell. Because you know friends, if the old cell cannot die, the new cell cannot be synthesized. What is the biological phenomena? The old cells or the functionless cells has to be replaced by the new cells. That is our rule. So what's happening now? If the damaged cell ORR, if the non-functional cell ORR, the old cell, whether the cell may be older, whether the cell may be non-functional, whether the cell perform all these functions, and now it has to undergo the biological process apoptosis. In case, even though it become older after performing all function, it is not dying. It is not dying. What do we can call? We can say that apoptosis is failed. Programmed cell death is failed, which means it is leading to the cancer. It is leading to the cancer. So now in this picture I am showing you three possible conditions. Everyone pay attention. In this particular slide I am showing you three possible conditions. Don't confuse, follow me correctly. Look into the first case. Don't confuse friends. In the first case, I will be calling this as the self-renewing stem cell. What do you mean by the word self-renewing stem cell? All the students look into this. This is cell having the ability or capability to synthesize itself. What friends? This cell having the ability or capability to synthesize itself. That's why we are calling it as the self-renewing stem cell. Now, the self-renewing stem cell give rise to itself at the same time. It is giving rise to the daughter cell. Now, you all people has to ask me a valid question. What is the question you all people has to remember? Does this daughter cell, can it give rise to the parent? No. So what is the statement I can give? I can say that this daughter cell is not self-renewal cells. Remember, I'm so sorry. The daughter cell do not have the ability or capability to give rise to parent cell. I am saying this. See friends, don't get confused. This is the parent cell can give rise to the daughter cell. But daughter cell cannot give rise to the parent cell. Now what the daughter cell is doing? Daughter cell will undergo the normal division. After normal division, it will undergo the process of normal differentiation. Now what's the point you have to remember? Then it is called as normal pattern because it is undergoing normal differentiation. Now after performing its function, obviously it will die. Hence what we can conclude? We can conclude it is absolutely normal. Very simple. Now friends, listen to me very, very carefully. Look into the second condition, don't confuse. Please look into the second condition very correctly. The second condition, please listen. This is what cell? Self-renewing stem cell. Friends, what has happened to the self-renewing stem cell? It is failed to give a daughter cell. It is failed to give a daughter cell. It is not at all giving a daughter cell. Instead of giving the daughter cell, the self-renewing stem cell on its own, it is dividing continuously. Obviously what? It is undergoing uncontrollable cell division. So obviously what we will be calling it as? We will be calling it as the cancerous. So second condition, there is a tumor possible. Why it is possible? Because the self-renewing stem cell is failed to give rise to the daughter cell. Instead of giving daughter cell, it is dividing on its own without regulation. Now the third one, don't confuse. Now if you look into the third one, don't confuse. Self-renewing stem cell giving rise to on its own, giving rise itself. And this cell, it is also giving daughter cell. And what we discussed that daughter cell should undergo limited division. Daughter cell should undergo limited differentiation. Finally, it has to perform its functions. But what is happening here? It lost its regulated division. It lost its regulated differentiation. It lost its normal function. Instead of doing all the regulatory mechanisms, it is dividing uncontrollably. As a result, friends, obviously it is going to give rise to what? Tumor. Don't get confused with the point. So, in the three conditions, only in the first condition there will be no chance of occurring tumor, because every entire pathway is normal. In the second condition, which cell got failed? Parent cell got failed, which means self-renewing stem cell failed to lead, which is leading cancer. In the third case, not self-renewing stem cell is normal. It has given the daughter cell also, but the daughter cell got failed. So they can ask the questions that when the chances of cancer occurring, whether the cancer occur when the self-renewing stem cell failed, or when the cancer occur when the normal cell failed to divide. So you have to remember what are the possibilities of converting, what are the possibilities of occurring a cancer when the self-renewal stem cell are working not normally, or are the normal cell working not working normally. Don't confuse. Self-renewing stem cell always works normally. If the self-renewal stem cell failed, it leads to cancer. Normal cell also worked normally. If the normal cell failed to divide, normal cell failed to differentiate, then only it will occur it will cause the cancers. So you have to remember the possibilities of occurring the cancer. Don't confuse with that. And friends, among all, the most important process, from here also we can expect the questions. What is it, friends? The process of metastasis. What everyone has to remember about this particular process of metastasis? Focus. Metastasis is not a big, simple step. Metastasis will occur in a multiple ways. What, friends? Metastasis will occur in multiple ways. Every student remember. In the metastasis, first, cancer cells are breaking away from where they first formed. Wherever they formed, what do we call them as? We call them as the primary cancer. From the primary cancer, it is moving to the other areas of the body. Then only we call it as the metastasis. Now the question is super valid. What is the question? Sir, how does a normal cell how does a cancer cell is moving actually? Now every student, please look into the diagram very carefully without confusion. Please look into this. This is the cell. Which cell it is? It is an epithelium cell. It is an epithelial cell. You all people look into the diagram. This epithelial cell, you can see the yellow color, right? It is protected by a basal lamina. Once again, friends, please focus. This epithelial cell is protected by what lamina? Basal lamina. So if that basal lamina is there, friends, the normal cell never break. But what happened? Since it is not a normal cell, it is a cancer cell. What is the specialty of a cancer cell? Cancer cell can show the process of invasion. So as a part of the invasion, it break the barrier, which means it break the basal lamina. Okay. After breaking the basal lamina, it has to come to the liver. But the question is, can it jump directly from the basal lamina epithelial tissue into the liver? Not possible. Now please focus. It has to travel from the epithelial tissue or the connective tissue to the liver. How is that possible? It is possible. It is traveling with the help of a blood vessels, or it is traveling because of the lymphatic vessels. So you all has to remember. For this only the scientist got the Nobel Prize in the year 2017. Remember, whenever a cancer cell is moving via blood circulation, what I told to all of you, out of 1,000 cells to move, only one cell can survive. What friends? Out of 1,000 cells to move, only one cell can survive. What is the reason? The reason, the major reason, lack of oxygen, lack of nutrients. That's what all, even scientists also will say that doing fasting at least once in a week is really good for health. Why? Because when we are under the fasting or the harvesting condition, the level of oxygen in our cells are low. When the levels of oxygen in our cells are very low, if the cancer cell wants to travel through the blood circulation, they require abundant amount of oxygen. They will be requiring abundant amount of nutrients. But we are under fasting condition. As a result, they won't get whatever they needed. As a result, friends, the cancer cells will die. Most of the cancer cells will die. But what is the disadvantage here? Even though one cancer cell survived out of 1,000, that cancer cell escape from the blood vessel, it entered into the new tissue, that is the liver, and it is forming multiple colonies in the new area. This is called as metastasis. So friends, in the metastasis there are two major steps are there. First step called as invasion, second step called as angiogenesis. Third only metastasis will occur. Okay, so friends, whatever we have discussed until this particular slide is the characteristics of a cancer. Okay, in the tomorrow one class, I will finish the causes of the cancer as well as the genetics of the cancer. Okay, see, from tomorrow also I will go little faster. By that, so we are having hardly 9 days. In the 9 days, my target is I have to finish at least 23 topics. So I will go little faster. Please try to adjust because we have already discussed once previously. Okay, so make sure that you revise it without fail. Okay, I hope the discussion is useful to all of you. If you have any kind of doubts, I am requesting all of you to ask. Clear, everyone? Please respond. Is it clear? It is very, very hot. The lights. Okay. I hope it's clear for everyone. Okay. Okay friends, that's all for now. We will meet in the tomorrow class. Okay. Thank you all. We will meet in the tomorrow morning class. Without fail, please revise the concept. Do not miss it. Okay. Thank you all. Thank you all of you. Bye-bye.